I'm here to introduce Dr. Emily Spellbrink, MD, PhD, from Stanford Children's Health. She's a clinical professor in neurology and neurological sciences and pediatrics. Dr. Spellbrink earned her medical degree from UC San Diego, and she completed her internship at Harbor UCLA. Her residency was at Stanford at Lucile Packard Children's Hospital, and her fellowship was at UCSF. Her special interests include genetic, early life epilepsies, and infantile spasms. She's also involved in PERC. Please help me welcome Emily Spellbrink, Dr. Emily Spellbrink. Thank you.
Good afternoon. Thanks so much to those who are here. I've kind of tried to design this talk as kind of how I would talk to folks in the clinic if you were here, trying to understand about a genetic epilepsy. I hope that will work for this audience, but if there are other needs or other questions, I'm more than happy to answer. Feel free if you want to interrupt or if you want to come up at the end, particularly if there are personal questions, please save those for a one-to-one kind of setting. All right, so I have no disclosures related to this talk. And I think before I waste an hour of your time, what's the importance of having this talk, of whether we're having a genetic revolution or some genetic confusion at this point with respect to pediatric epilepsy? The reason I think is that it's a question that's here to stay and something that's very important with respect to epilepsy. And we learn that more and more. So genetics actually causes 40 to 50% of our difficult to treat or early onset epilepsies, and 27 to 70% of our neonatal onset epilepsies or developmental and epileptic encephalopathies. That's a lot. In fact, for this reason, it may come to pass before too long that we actually have a recommendation for exome sequencing as first line in epilepsy workup. And so understanding something about what that means, what it means for our children is going to be very important, even though it can be a confusing subject. The impact is clearly rapidly increasing and here to stay. We have pivotal treatment decisions that result from some of these genetic diagnoses. We know that in a lot of cases they can help with prognosis and with family planning, and we are hopeful that they will give insights to precision medicine therapies, whether those are adapting drugs that we already have or whether that's actually directing a gene therapy towards your particular epilepsy. It's very helpful to have this information to be able to work toward that goal. But does it actually lead to just more confusion? So in general, I find that when my families get these results, they're staring at their cell phone, kind of like this gentleman here. There are pages full of small print with confusing letters and numbers of genes that no one's heard of. There certainly can be problems with specific labs that have analytical failures and lead to misleading results. There can be variants of unknown significance that are uncovered. And in those cases, different doctors can give you different perspective about whether those are important or whether they mean something for your child. And the understanding of whether they do or not may actually change over time as our science changes. So all these things can be very confusing and can cause a lot of kind of emotional ups and downs in our understanding of the disease. There are also unintended discoveries that can come from genetic testing. So in some cases, non-paternity is discovered. In other cases, incidental findings about genetic risk for things other than epilepsy can be uncovered. And, we try to talk this through with families and give you some choice about what you may or may not want to know, but certainly those could be unforeseen consequences. And maybe even understanding sort of the implications of your genetic results is another difficulty. So, what are scientists doing to study this if my child has this disease? Does this diagnosis lead to any different treatment options for my relative? And certainly there's the fear that there may a diagnosis may introduce some false hopes or false understandings. So the goal of this talk is really to try to help equip you with enough basic genetic understanding to understand this revolution a little bit and feel like you can really be a part of it. Additionally, I'd like to try to minimize the confusion and false hopes that are kind of lurking out there with respect to genetics, and then to introduce some excitement for the real advances that are coming in this field, and some hope for those of you who may have genetic diagnoses, for you or a loved one. So overview, I'm going to try to start with how can genetics lead to epilepsy. This is also called my my genetics, epilepsy, and Legos talk. What is the role of genetic testing in epilepsy, the kind of common types of tests that you could be offered, and what the potential yield of those tests is in different individuals? What are the possible genetic testing results that you could get? Do I just get only a result if they know exactly what my epilepsy is, or what else could I find out? And then what is the possible impact of those genetic testing results, which I hope will also introduce some hope for the future. Starting with, how can genetics lead to epilepsy? So let's define our terms just briefly so everyone's on the same page. Epilepsy is an enduring predisposition to generate epileptic seizures, meaning I don't have to clock you over the head for you to have a seizure or give you a drug. You could just have it because of the way that your brain is. This predisposition is usually due to an abnormal brain structure or function that's causing abnormal electrical communication. Our genes encode molecules that are responsible for brain structure and function. And so if there's a change in the genetic code that interferes with the brain structure or function such that it predisposes to seizures, you could imagine that a genetic epilepsy could result. How is the genetic code stored? So again, this is very basic overview. Can you guys, yeah, you can see my arrow. Okay. So each person, right, is made up of many, many cells. And inside each cell is a structure called the nucleus, in which all the genetic material, the code for making that person, is stored. And I'll say that with a little caveat because there are some other genomes and mitochondria and other things like that. But for the main purposes of this talk, the material that we're going to be talking about is in the nucleus of cells. Within that, you can imagine it's a very, very big code to make all the proteins and structures that make up a whole person. And so the code is kept in this very tightly wound material, which is in humans into 23 pairs of chromosomes. And if you unwrap each of those within that, you can see that there are all these base pairs. So this is the actual code. Like within, if you unwrap and unravel a lot, you can see all these actual nucleotide pairs of C's and G's together, A's and T's together. And so when you get those results that come from the genetics company, it will have your which kind of letters of this code are changed, or which segments of this code are changed in the particular individual who's being evaluated. And that's a lot of what those letters and numbers actually are. So if you want to envision how they relate to a structure that's in a human being. How is this genetic code used to make you? So this is my genetics and Legos portion. So on the left side is all the nice biology diagrams of how this works. And on the right side, I'm hoping, is a little bit more user-friendly understanding of how this happens. So up at the top, on this side, we can see that there's the code itself, and there's a way that we kind of translate this code into instructions for making proteins, the building blocks of the body. And so over here, this is quite similar, right? We have instructions of how we need to make our Lego model, right? And the instructions have two pieces. One piece is they have to actually show how to make the Legos themselves, right? Like what shapes do we need with how many little bubbles, and what colors. And then on top of that, we need to know how to put the colors and the shapes together the right way to make the ultimate big picture. So you can think of the transcription process as like how to make from these plans into actual Lego blocks. Like what sizes and shapes and colors do you need? And then you can think of the translation process where the actual proteins are made as the actual making of these Lego blocks. And then the final step is when the Lego blocks actually get all put together into structure, and that takes interaction of many proteins and systems within the body. So that's kind of how it works from the ground up. I hope that made some sense. Any questions on this part? Okay. What can go wrong with the genetic code? So many, many things can go wrong, and this is certainly not exhaustive, but the big categories we tend to think of is you could have extra or missing whole chromosomes. The standard example we think that's most common for this is something like Down syndrome. There's a whole very large extra piece of genetic information that's present in what's called a trisomy. And sometimes a whole extra piece or chunk can be missing, and in that case, it's called a monosomy. And there are variations of that. So extra whole or missing chromosomes, big pieces of code. The second is extra or missing chunks of DNA within a chromosome. And when you get just a chunk that's missing or extra, it's usually called a CNV, or copy number variant. And these are the kind of things that are picked up on a microarray test, or a SNP test is sometimes called. And then finally, errors in the actual letters of the base pair code. So this is once you really unwrap everything and get down to the nuts and bolts. Maybe normally this should be an A and a T to make the protein correctly, and suddenly it's not. It's mistaken and switched for a G and a C, and that causes trouble in the actual structure of the protein. And so that I think of as, that's the kind of thing that a gene panel or an exome or potentially a genome is going to pick up. And you can see over here, it's the difference between, like a normally formed Lego and maybe a Lego that's not quite structurally soundly formed based on a difference in the code. And then we think about, well, what implication does this have? It can have really broad implications, right? So sometimes when your code doesn't work and then your molecule doesn't work, you can get big structural changes in the brain. You can get things like tubers and tuberous sclerosis. You can get things like lissencephaly or smooth brain disease. And all those things are types of changes genetically that can cause seizures. And then on the other side, you can also get a brain that looks completely normal, but it doesn't work right. So you can have ion channels, for instance, that aren't properly made. Those are the electricity conducting parts on the cells. And when that happens, again, you could certainly get an electrical storm in the brain. And there are many other types of molecules that can go wrong besides just the ion channels. There are cytoskeletal proteins that help to make and transport things in neurons. There are solute carriers, so things that help to carry around important molecules in the cells and or do metabolic processes in the cells. There's cell cycle regulation molecules, so things that regulate dividing of cells. And I won't list everything here, but you can imagine there are a lot of different things that could disrupt the function of what neurons normally do and how they conduct electricity. And each of these categories has numerous examples of how they can cause genetic epilepsies. So what is the role of genetic testing in epilepsy? If we look at the causes of epilepsy, and again, this is slightly outdated. I think we have even a slightly better knowledge now. By far, the majority of causes, when we're able to find a cause, are genetic causes. And when we think about genetic testing, who do we think about doing genetic testing to? So for instance, one might imagine if you've had a very large brain injury, and that's clearly also the place where seizures are coming from, you might not think to do genetic testing for that person. But there are also certain kinds of epilepsies that even if you don't have a family history, you may want to get genetic testing on. So I think what we think about is whether the affected person's story kind of fits a known epilepsy syndrome. For almost anyone who has very early onset epilepsy, like less than 1 year old, those folks will be thinking about whether there could be a genetic cause unless there's a very clear other cause. For refractory seizures, epileptic encephalopathy, particularly with other workup unrevealing, we almost always think to do genetic testing. For mixed epilepsy, which is a kind that is not just coming from one place, the seizures, and it's also not just coming from the whole brain all at once, but there are some components of both, makes us wonder whether something's wrong within the brain more diffusely and whether we should check for a genetic cause. So that's a mixed epilepsy. For epilepsy plus kind of syndromes, where in addition to epilepsy, there's autism, other developmental or congenital anomalies, we wonder whether there's something that could be wrong with the code. And then, of course, for folks who have a really strong family history of epilepsy, we always wonder whether there could be a genetic component. Now, what about if you don't have a family history of epilepsy? Is there any point to even getting genetic testing? I don't have epilepsy, why genetic test my child? And I wanted to try to kind of make some pictures that help explain this. So sometimes we find that parents may have a gene that could predispose to epilepsy potentially. So I tried to use my Legos again here. So this side of the Lego bridge you can see has a very nicely formed, normally formed purple block. And this side has a purple block that's missing just a little bit. And what's going to happen to this bridge? Is the bridge going to break or is the bridge going to hold, right? So if someone has a gene that's maybe an unusual variant that could predispose to epilepsy, there are a lot of things that determine whether it actually does end up being epilepsy or not, besides just the gene itself. So perhaps if we just left this bridge by itself and didn't put a lot of heavy things going over it, the bridge would hold and we would not diagnose an epilepsy. But if other big stressors come, like bad brain repeated concussions, or certain illnesses or diseases come, they put extra weight on that bridge, right? And they stress it out. So the parent may have not had a certain exposure or insult that a child does have, and then the child may end up expressing an epilepsy when they really have the same gene as the parent that the parent has not expressed. So those are environmental factors that can influence. Second is a polygenetic influence. So one could imagine that if all that is changed in terms of bridge structure is just this one little piece of the purple Lego block, maybe the bridge holds fine. But once we start chipping away at other Lego blocks, moving things down a little farther, the bridge weakens further, right? So this is how, like, if you have a different set of genes combined together, they make a different bridge altogether, and you can see a risk that you might not see in someone with a different combination of genes. There are recessive disorders in which, perhaps each mom and dad have one normal Lego block gene and one not functional Lego block gene. And both of them are okay because they had one normal one. But when two of the ones that are not working are both coming together in the child, then that child would manifest epilepsy in a way that the parents didn't show in either one of them. And finally, there are mistakes that happen, in the copying of genes somewhere between the making of the parent and the full making of the child. And those are either somatic or they're de novo variants that can be either somatic or germline. But in some cases you can imagine. So if, let's say, the brown color here is the affected person, the person with epilepsy. So in some cases we might have a parent who has epilepsy and then they have a child with epilepsy, and that's kind of a dominant condition that we understand how it's inherited very, very directly. But what if dad actually has, or mom actually has, a change in just the gametes, just the cells that make the baby, or just some of those cells that's different from all the other cells that make up dad? Then those could be passed on to the child, and the child could just come from one of those germ cells or gametes and could have a gene that expresses only in the germ cells of the dad, the sperm of the dad, but not ever in his brain to make him have epilepsy. There are also things where the child from embryo itself, as he or she is dividing, that there could be an error, a mistake, or a change in the genetics at that point in time, in which case the child would look different from the parents. And then there also can be somatic mutations even within the child. So as the child is dividing, perhaps there's an error fairly early on that only affects brain or only affects certain parts of brain. And in those cases, we might see a child with unaffected parents or other family members. Common types of genetic epilepsy. Did you have a question? No, okay. Common types of genetic epilepsy testing. So often the first line or more general one is something called a chromosomal analysis, sometimes plus minus including a microarray analysis. So chromosomal analysis is just literally looking at and counting the numbers of chromosomes, making sure if you have an X and a Y if you're a boy, an X and an X if you're a girl, and the total 23 pairs. And if you don't have that, then that by itself could be an explanation for why you have some differences in how your body works. The smaller version of that is looking for those copy number variants we talked about earlier, which is just within this chromosome, like some little chunk is either duplicated or missing or triplicated. Those studies just kind of look for larger pieces of the DNA that have been changed. The second kind, which also often can be first line for folks with epilepsy, is called a gene panel. And it's taking a lot of common genes that cause a lot of the epilepsies we know about and just screening those and saying, hey, is any of these common ones that make epilepsy for a lot of people, what's causing epilepsy for you? And so when you get, I've had a lot of questions about this from my patients, when you get a gene panel for epilepsy and it doesn't clearly show us what causes your epilepsy, that doesn't mean that you can't have a genetic epilepsy. It just means that you don't have one of those somewhere between 10 and 600 very common genes. Different companies have different numbers that we know cause epilepsy for a lot of folks. So it doesn't mean that workup has to end there. And for some folks, it does mean that workup ends there because you get an answer. But if you don't, it doesn't mean that it can't be genetic. And then usually second line, but perhaps to come first line, based on some recent data, are these other things, a whole exome or whole genome sequencing. And this is actually looking into the code, the A's, C's, T's, and G's that are the code making up the person that are embedded within all this genetic material. And that tends to be the more difficult to understand stuff, and the stuff where you get answers about things that aren't only seizures, right? So the codes of what makes up your endocrine glands and the code of what makes up your kidneys, and all those things are also in these larger kinds of sequencing. And so some people want to know if something is found with respect to non-epilepsy answers, like, for instance, risk for Alzheimer's disease. Would you want to know that information if someone looks at your whole code? Versus others would not want to know that information. They just want to know whether or not something related to epilepsy is found. And then there are also special cases of certain kinds of epilepsies that we know have other genetic ways that they're transmitted. And so there's specific tests that might be done if something is suspected based on the way that the patient presents. So common yields for these studies, the one that looks at whole larger chunks of DNA, pieces of chromosomes, tends to be somewhere between 5 and 13%, finding an answer for what might be causing your epilepsy. For epilepsy gene panels, so that's the one where we take a bunch of the really common genes that cause epilepsy and look, that tends to have 15 to 38% yield in different studies. For whole exome sequencing, which is often done as a trio with the patient and the patient's parents, we get pretty widely vary. So, almost 2 times varying how often you can get results, 30 to 60%-ish. Mean is about 31%, and the range 8% for kids who also have autism spectrum, especially higher when we strongly suspect that there's one gene that might be causing the disorder. And then exome alone, we know this is part of why this may become first line. Exome testing by itself will exceed the yield of a conventional workup for epilepsy. So we know this can be really important in finding out causes. And then especially for folks who have these kind of early onset or developmental epileptic encephalopathies, first year of life, or with certain features of how it also affects your cognition and your development, we really try to assess these because about 50% of those will have a genetic cause. And if we take not only the part of the genetic code that codes for proteins itself, but also the part that regulates how that code is read, if we take the whole genome altogether, then it yields an even higher percentage diagnosis for those folks with really early presentations. So what are the possible results when we get genetic testing? And I'm going to simplify this somewhat because there are really a lot of results one potentially could get. But I think in terms of big categories, we think of benign, which means we don't think it should negatively affect your health in any way. Pathogenic, which means we think it should cause disease. And variant of unknown significance, which means we're not really sure if this causes disease for you or not, and we need to think a little bit harder about it and whether it could be important. I would emphasize that when we get a variant of unknown significance type result, it really should not be used as a positive result without additional supportive evidence. So if you have an unknown variant, unless we have reason to believe that's really what's causing the problem for the patient, we shouldn't be treating with directed treatments based towards that. And I'll give you, again, kind of analogy-wise and Lego bridge-wise, how I explain this to folks in clinic. So If we think about variants of something within the population that we're all very familiar with, you can think of eye color. We know that many people in the population have blue eyes and many people in the population have brown eyes. And as far as we know, those particular eye colors don't really have any significance on vision. People with blue eyes and brown eyes all should be able to see okay. Some of us need glasses and some of us don't, but the eye color itself doesn't cause our vision to not function. Then we know that there are pathogenic variants of eye color that actually do affect vision and eye safety and disease. So, for instance, albino eyes, which are lacking in pigment and look kind of a little bit reddish even, let too much light in. And having too much light let in does not give a clear image for vision, and it also can even cause sunburns on your retina and other things like that. So we know that that's a kind of eye color that actually can cause problems for your eyes. So if you think about these two with respect to genetic variants, I would say blue eye, brown eye, those are all examples of benign variants. An albino eye is an example of a pathogenic variant. We know something bad can happen from it. Then there's violet eyes, right? So they're rare. Not many people have violet eyes. Maybe nobody's ever made a paper about whether violet eyes cause any problem with vision before. Maybe we don't know what happens when you get violet eyes. So we can use some things to try to model and predict, but we're really not sure in the end whether violet eyes is an eye color thing that could affect your vision or not, but it is a result that we could get back if we do some checking of variation in eye color. So that's kind of how I think of variants of unknown significance in genetics. It's like violet eyes. And here's the bridge example. So, again, a benign variant, we expect the bridge to hold, right? A pathogenic variant, we know that there's such a big piece missing of the bridge that there's going to be a problem as a result. It's really very unlikely that the bridge will hold and not cause trouble. And then a variant of unknown significance, well, there's something slightly different about this bridge, but we don't know if it's going to hold or not. And maybe it needs some more study, and maybe we need to think about structural dynamics of a bridge and model it and things like that. And so this is very much, a spectrum, and I think that this rainbow shows it very nicely. We have a spectrum of benign to pathogenic variants. There are some variants that are really strongly associated with causing trouble, and we really know that they're going to be responsible for epilepsy in most cases. And then there are variants that, I went on the wrong side here. This is the strong ones. And then we also know that there are some that we're very suspicious are not going to cause any trouble. And then there's a whole bunch in between where they might introduce some risk, and maybe in the right circumstances, they could lead to epilepsy, but they might not always. And this is what makes things very difficult to interpret, particularly in the context of a parent who has a variant and is not affected with epilepsy, and a child who has the same variant and is. So I think we tend to think of these outcomes as benign, the good, pathogenic, the bad, and variants of unknown significance, the ugly, because they're not a neat explanation. But I really don't think it's quite that simple. And I wanted to highlight, what is the possible impact of genetic testing results. This was a Wall Street Journal article from a few years back. The unfulfilled promise of DNA testing, rapid advances in genetic testing, whipsawing families' diagnoses and treatment. And this was about a little girl who had a variant of unknown significance, was counseled, according to the article, by an MD that this was the cause of her epilepsy. Family became very invested in that diagnosis and in the community. Later, with further scientific advances, it came out this was likely not the cause of the epilepsy. Of course that's a whipsaw for a family, right? You think you understand what's going on with your loved one, you've committed to a community that's helping and supporting you, and suddenly that's not the case anymore, and have you even been doing the right thing for your child, right? And so I think this is what leads it to not being so simply the good, the bad, and the ugly, and also leads people to wonder if they should do genetic testing and to be a bit mistrustful. So I'd like to highlight, just so we all kind of are on the same page about how this goes, benign. Can it really be the good? It is often the good, and that excludes, if you get a negative, like a benign panel, it excludes a lot of really bad causes of epilepsy. It means that we've looked for, in the case of, say, a gene panel, we've looked for a bunch of genes that we know cause epilepsies, including really bad epilepsies, and that's not what you have. So in some ways, a benign result is a good news, but it also leaves you without an explanation for your child's epilepsy. And it could occasionally be misleading, because what if, the lab has actually just not done their due diligence with how they've done the studies and you've missed something? Or what if you take the result to mean that therefore I don't have a genetic epilepsy, but in fact all you've done is a gene panel, and you might have a genetic epilepsy that could be shown with other kinds of testing. So you have to keep these kind of caveats in mind with a benign result, and continue to work with a good trusted provider to understand what actually the result means and what the next steps should be. Again, pathogenic, we think, oh, we found something bad, right? This is the bad. But in fact, we've gained information. Usually it suggests that something is wrong, and we may be able to use this to predict what might happen in the future, to make decisions about family planning, to make decisions about whether there are other things in terms of health information that need to be looked at. So for instance, children with tuberous sclerosis, once we find out that that's the cause for their epilepsy, they also need to be checked in their skin and in their development and in their kidneys and in their hearts. And we can prevent other problems that might happen because of this good knowledge. And then the kind of ugly piece. So usually getting this pathogenic variant doesn't actually give you precise prognostic information. So for instance, the gene SCN1A, I'm sure is well known to many in this audience. It can be present in children with simple febrile seizures who outgrow them and go on to develop completely normally. And it can be present in children with Dravet syndrome who have devastating epilepsy and developmental disability. And even within Dravet syndrome, children do a variety of different ways. And in the end, they're still your child, right? They're still the same person that you've grown up with and loved before that genetic diagnosis. And now having this information may just make you worry more. But you have to kind of keep in mind that we don't have a crystal ball, even with a genetic diagnosis. And there can be a variety of outcomes, and you will always be the one helping your child to come to the best of those outcomes that you possibly can. Finally, the variant of unknown significance. So is this really the ugly? It certainly can be confusing, and it can lead to misguided actions or beliefs, particularly if you don't consider it to be something like the stock market, where we're weighing the probability that this is or is not responsible for the epilepsy. If we take it as absolute truth that it is or isn't present, that's not what it is, right? It's a variant of unknown significance. Now, it is hard to live with unclear significance, and so that can also be taxing. But on the other hand, if we find a variant of unknown significance that's interesting to someone, it can really lead to good opportunities for research. It can lead to other community with other people who have variants of unknown significance, and how can we get these studied for our families? And it can also, over time, be reanalyzed as year to year more information comes out, and it may lead us to either a diagnosis or a clearing of that variant and knowing that it's benign. So I think, why would we want to go through all these things that, all this uncertainty, all this unclarity, all this potential for ugliness? Certainly if we get a diagnosis, and if that's something that we really want, psychologically and for family planning and all those reasons, that's a wonderful thing in and of itself. But I think the real holy grail with this revolution is to gain a precision medicine approach to the care of our loved ones. And so the things we can do with precision medicine at this point is, as I mentioned, we can identify additional medical care that may be needed to prevent harm and prevent unwanted outcomes, such as in the tuberous sclerosis example, screening the heart, screening the kidneys, getting advanced set up for a lot of developmental support, as we know children are at risk for delays in autism, really getting good access to resources early on, preventing SAGA complications in tuberous sclerosis, where they have tumors that grow and can shut down the ventricular system from running and cause the head to swell. So all these things, like once we know that information, we can work on. We can also identify the most relevant support services. So you would get access to families with other children who have the same kind of epilepsy, the same cause of their epilepsy, and they can really provide useful tips as well as just emotional support and resource support that they've found helpful. We also know that with these diagnoses, you can again get early developmental access to resources, and that can improve outcomes. And then finally, for some genetic changes, we actually have targeted seizure medications that we either should or should not be using. In the case I gave of SCN1A pathogenic variant and Dravet syndrome, we know that depending on whether it's a gain or loss of function change, but for those who have a loss of function, we tend to avoid using the sodium channel blockers, and we know that it can make it worse if we use those medications. It's not true for everyone with SCN1A, right? And we have to think about what that change has actually done, but we do know that that for some folks that's the case, and it can also lead us back to this research part down below that we could potentially even study the specific change that's in a person and ask what is good for this or what is not good for this. We also have specific diseases that we've kind of used drugs that are already existing to specifically target, such as memantine for GRIN2A, and I'll show you that in just a moment. We can kind of think about, could we use a medication that we already know of that would really target this mechanism of epilepsy? And then, of course, the best thing we'd like to do would be able to fix the epilepsy itself. And with drugs, just straight treatment drugs, not any kind of genetic therapy, this has been started to be explored with the PREVENT and EPI-STOP studies for tuberous sclerosis, using vigabatrin even before the onset of seizures to see whether that can alter, and it seems to be able to alter seizure and epilepsy outcomes for those children. That's kind of our first pilot example on that particular thing. But of course, it leads us also to wonder about gene therapy trials. And these are no longer pie in the sky. These are actually occurring right now. So the Tango and also the Monarch trials for Dravet are all going on. I put in a plug for some of you might have heard Dr. Numis talking earlier today about genetics, and UCSF is actually running a couple of those trials if anybody's interested in getting involved. But there are also targeted medications in development, such as potassium channel drugs for those with potassium channelopathies, particularly the gain of function ones, and for characterizing new diseases as well. So in some cases, an interesting variant will be of interest to researchers on the disease, and they'll actually express it in cells in a dish and test different compounds on it and see if it affects how I'm thinking of ion channels in particular, but how those can conduct ions in and out. In other cases, it may be that you can contribute either phenotype information or actual samples to a natural history study that's going to understand what people with various variants of this gene can look like. And it may help it to become clear that this is what's causing the problem for your family member, or it may also help to clarify that there are some cases in which this gene doesn't cause a problem. And then either way, you're contributing to your understanding as well as that for the whole community. And then finally, I wanted to point out that just the rate at which this is happening, so I know it sounds like, well, maybe you can contribute to future knowledge, but this is really happening quickly, and it's happening now. Trio exome and genome sequences can now identify the causative gene in epilepsy. 63% for, again, those early onset developmental epileptic encephalopathies, and just 4 years ago it was only 32%. So having this iterative process here where our variants are contributing to knowledge and understanding and research, it's really moving things forward. So it's not just like, well, maybe if I give my information to do this, it'll have some impact on someone in 50, 100 years. This is happening now. And I think it's really wonderful how many people have lived with some uncertainty as that being the risk, with the benefit being that they've really contributed to some understanding for a lot of families. So I'll show you this. This is the example I had talked about. Can we target medicines even that we already have once we know a genetic change? Great example of this is a GRIN2A mutation or a pathogenic variant in memantine. So memantine is a molecule that binds in the glutamate receptor and has been able to help with people who have epilepsy caused by this being too open. So it kind of helps close things down and kind of allosterically modulate. And then you can see the EEG on this. This was just a case report, but went from being very abnormal, lots of epileptiform activity, to looking relatively normal, and correspondingly the seizures improved greatly for this patient. So if you happen to have something that would be amenable to that, genetic information will let you get there to trying out that therapy. Another one I will give is drugs in development for CDKL5 deficiency disorder. So this was, I made this slide, gosh, as you can see, 2019, right? So this was the clinical trial status when this excerpt was taken, right? And then I've tried to update it for what's actually happened. So for instance, these were all substances that were in trial for folks with CDD, and they were, as of 2019, they were active recruiting, and now we know that ganaxolone actually does help with a decrease of 31% in major motor seizures for folks with CDKL5 deficiency. And then there are several others on here as well, with some positive results for soticlestat, although again early, not like a formal clinical trial, but I gave you the information, as we currently have it. For fenfluramine as well, seems to reduce seizures for these patients. So a lot of good initial promise, and this is only within 2019 to 22. So if you have this information for your family member, it really can be helpful in a relatively short period of time at driving things forward. Finally, I'm sure of interest to many is the current kind of gene therapies that are starting up. And does anyone in here interested in gene therapy for a family member or a friend? No. Well, I hope maybe at least interested to understand it. The trials are just beginning, but again, for the SCN1A that I mentioned, that can cause everything from simple febrile seizures all the way to Dravet syndrome. Folks with Dravet syndrome really have a lot of challenges when that manifests badly, right? They have a greatly increased risk of sudden unexpected death in epilepsy. They typically have neurodevelopmental concerns that go along with their epilepsy that affect everything from their ability to interact socially, to learn, to speak, and to walk. And so for this reason, that this is a really highly impactful disorder, not even to touch on the intractable seizures in epilepsy, it's been one that we really would love to target and get rid of prophylactically, right? We'd like to be able to not have that genetic predisposition anymore to all these problems. And so to explain very simply, there's this method called TANGO that has been used. So it stands for Targeted Augmentation of Nuclear Gene Output. And it's technology that allows development of an antisense oligonucleotide. And I'm not going to go into a lot of detail about what that is, but I would say, generally speaking, it's kind of something that can direct the code to either be made into a protein or not made into a protein. So I'll put my little Lego example up over here. So if we do things normally, we have a gene that's a code that's being expressed. This is the plan for, hey, we're going to make a protein, okay? And then here is the actual, like, this is going out to be made into a protein, right? And here's the protein. The code getting expressed of we're going to make a protein sometimes is made more than is actually needed. And so the extra copies that are made are sometimes have a specific sequence that says, okay, like, let's get rid of this one. We don't need quite so many. But one might imagine that if you have, so here's your two genes, right? One from mom and one from dad. And you have one from mom that makes normal red Lego blocks, and that's what you want. And here's the extra stuff that's going to get broken down. You also would have extra stuff that could get broken down from this one, but this makes the faulty Lego block that's not going to work so well. If you were to be able to get rid of this, one could imagine that you'd have more Lego blocks that work well, but also, even if you can't get rid of this faulty Lego block, if you kind of drown it out by so normally we'd be getting rid of these extra red Lego blocks because that's just how the system works. But with Tango, we take away getting rid of that, and now we would have twice as many normal functioning red Lego blocks. And so hypothetically, we could kind of drown out this not working very well Lego block, or at least add some more of the good working into the mix, right? And so this is the idea that we want to add more of the functioning SCN1A channel into the mix and kind of drown out the one that's not working so well. In mice, so far, intracerebral delivery of this has decreased the death associated with Dravet SCN1A, and also reduced seizure frequency and severity. And so this is now in clinical trials, early clinical trials with humans. There are many other possible targets of tango, and I put this on as an old kind of prospectus list of things that people thought would be amenable to tango if this works out well from SCN1A. Of interest, UBE3A is also has the same mechanism in a clinical trial, so this is the Angelman's gene. But there are certainly still a lot of challenges related to this. So certainly getting the dose right. So if we take away or add extra copies, we still want to make sure that it's not, you don't want too much or too little SCN1A activity, and we have to figure out how to get that right for folks. Delivery of the antisense oligo to the brain is a challenge. That is something people are solving in different ways for different gene therapy questions. Human safety and efficacy, of course, are key to us wanting to use any of these therapies. And I put in here several of the things that are, like when we're in mice, we're not as devastated if they have a devastating immunologic response, or if we end up modifying other genes in some way, but with humans, we're really quite worried about those things. And so even things that have worked previously, like using CRISPR modification, stuff like that in other animal systems, are not quite ready for prime time yet, although there's certainly hope that these things will come to pass in the future. So I think the key to making this whole genetic mess a revolution instead of a confusion is to understand the limitations and uncertainty that are associated with genetic testing, not to take any results that we get as this is some sort of gospel truth that's the only thing that can be true for my family member, but to kind of talk through and interpret with care with a trusted team what this information means and how much of our faith and certainty and treatment we should really be basing on it. And then we, of course, want to use our genetic testing to the best of our abilities to advance our knowledge of epilepsies and to advance therapies for our epilepsy patients. And that certainly comes as a side benefit that hopefully impacts your loved one as well as many others. So I leave you at the bottom with this is our epilepsy team. There are many, many resources for those with genetic epilepsies. I put a bunch of their icons on here. Many of these groups are here. And I also wanted to highlight Dr. Knowles, who's one of our epileptologists that I just showed on the previous slide and will show again. Was the first author on a paper from this year that highlights a lot of resources. I don't know if anyone wants these, please feel free to take a screenshot. But I think this is a lot of different organizations that you can go to kind of get some guidance about this and to connect with other people who might have similar concerns, interests, questions, and all those kind of things. And with the groups that are driving the research in case your provider is not getting you hooked up with those folks. So here we are. Thank you very much. Feel free to reach out with questions to me, and hopefully you guys found somewhere there's a bunch of stack of paper with my contact. And then also we're always happy to have genetic folks come to our genetic epilepsy team. And this was my daughter from last time we were in person here. She's now like 6, but it shows you how much can happen in 3 years. Thank you. Questions?
What about mutations? Is that the same as, because he had a mutated gene, is that?
Yeah, let me go back to that part. So I think the term mutation has fallen out of favor in terms of politically correctness, so that's why you didn't hear me use it too much. It's the same as a pathogenic variant of mutation. So it's just, instead of saying, because I think people think if they hear that word, well, then my child's a mutant or something, but that's certainly not the case, right? I said there's a spectrum of everything. So there's changes that we're pretty sure cause disease, and those were previously called mutations and are now sort of pathogenic variants. And then there's ones that we're pretty sure don't, and those are the benign or, benign variants are like a negative study where we didn't find a result.
And how about like doing stem cell? Has any of that in fixing any of this, or?
So there hasn't been anything so far where our stem cells have been used to treat or reverse a specific genetic condition, but certainly stem cells are being used to study many of these genetic conditions, including both human and non-human stem cells, and including organoids. I don't know if you've heard of those, where they've actually gotten like brain cells that they get to come together and form little mini brain connections in a dish. So it's very useful if we know the specific change that your child has, to be able to cause that to happen in cells in a dish or in stem cells, and then to study things that way. But so far, as far as like putting the stem cells into the brain and seeing that correct a genetic problem, that's not available right now as something that's in clinical use. I know people do put stem cells in various places in the world without much good evidence to support that that's going to be helpful.
Dr. Spellbrink, how should we handle all of these variants of unknown significance? There are plenty of companies that will sell you a genetic test, maybe build insurance, maybe not. And what are they basing their, if they're a pathogenic variant and it's in the literature and everybody knows that it causes disease, that's one thing. But we're discovering all new variants all the time, and they just get shoved into this wastebasket of the US. And there's no like standardized way to adjudicate which of these VUSs are really high risk and which are probably just benign variants. Is there a proper way we should be doing this?
Yeah, so I think that's a great question. And I also, what I thought you were going to say is there a way we can make kind of a standard pipeline of investigation with VUSs? That's a great question, but would need a lot of funding. But I think the underlying question is, how do we start to evaluate whether something is significant or not? And I think the ways that most of our companies do it, they do try to help us out, right? So the companies themselves will often offer genetic counseling, and then certainly many institutions will also offer our own genetic counseling. And we look at several things. So we look at modeling, like if we change the code in this way, how do we think that that's going to make a protein? We can use computers to model this, right? So that whatever happens to that protein, we can kind of get a sense of like, does it look like it's just going to be pretty much the same protein, but maybe with some slight wiggly part on one side, or is it going to be really a different protein that's not going to work right? So that's one way we do is with in silico modeling, right? And then we also do looking at population databases, which means when we've looked at like lots of different people for genetic studies, but not specifically looking at epilepsy, does that particular change just come out in people who don't really have epilepsy, that we're not studying for epilepsy, but they seem to tolerate it okay? Do we have any information about it from preclinical studies, like things that we've looked at where someone's changed a gene that way in a mouse, or where someone else has reported it from another lab, right? Another person with epilepsy features has that same variant, then that starts to build a case for us. So all these things, and whether someone's done cells in a dish, all these things. So these start to be ways that we evaluate. And I know you know all those ways, Dr. Nguyen. So were you getting at anything other than that, or?
Well, there are many ways to do it. I just don't know how it should be done.
How it should be done.
Like, should we be forming consortiums, international consortiums, to talk about all these variants that are coming up? Should we be forming registries of all the variants that are out there, but then you get into like the private databases of these companies that are selling these tests to begin with?
Yeah, so the companies definitely they have some degree of their own responsibility where they have to reevaluate data every few years and come out with whether now we know that this is likely benign, likely pathogenic, or for certain pathogenic or benign, and update the ordering persons or families. But I think, in addition to that, there are a lot of people handling it different ways. So I know the Pediatric Epilepsy Research Consortium, for instance, is garnering a genetics database, but within that database, they're not only including pathogenic variants, but also the variants of unknown significance. So we'll have a relatively large, non-proprietary sample there that hopefully can be mined for study. And if you collect enough numbers, hopefully we'll be able to, across the different genetic companies, we'll be able to actually get a little bit more information.
What about us that know what our child has, but we don't know, like we've only been told, well, there's, you know, X amount of people in the world that have this, but we're not told they exhibit the same things. Like, you know, what if that person is doing something that can help him? How come we're not told?
Are there, so you've been told that there are other people with the same problem, but there's no information to be shared from those people?
I have not been told.
Okay, why don't you come up after and let's talk, okay? We'll see what, because I think that that's a very circumstance-specific kind of question, and of course that would be very hard, but how that can be answered for different diseases is a bit different. Okay, so talk to me after. I'm sorry about that.
Yeah, what's the greatest barrier to your work?
It's so much beyond my work. I think, you know, I listed some of the logistical barriers in terms of gene therapies going forward. But I think in addition to that one, you know, we talked quite a bit this morning about those things with our partners in industry. And I think that being able to study things that are relatively rare and that cause a spectrum of how the outcomes are is difficult. Because we don't have the best definition of exactly what outcomes do we expect. I mean, we can do seizure counts, but what if, you know, within the range of what this causes, people can have rare seizures or really frequent seizures? Then we can't just count how many seizures a person has in a month and decide whether it's, you know, like whether they've had a change in how they're doing, right? So I think that, and then also the developmental aspect, right? So many families find the developmental challenges to be even worse than the seizures, and those plus minus relate to the seizures as well. So if you have a lot of seizures, of course you have problems with your development because you're busy doing other things and being in the hospital and all that, right? So trying to have a kind of outcomes measures that parse all those things well enough to be able to say, hey, we made an intervention and this really did something. I think that's a very important target that kind of academic centers are more working on, because it's not necessarily with respect to only one drug or only one genetic change. It's like we have to find ways to do this in a larger, more systematic and better way. So I think that's one of the main challenges. And trying to get funding then to be able to do that is obviously a challenge. Is that what you were thinking?
I was just curious.
Well, thanks for the question. All right, thanks everybody for coming.